Well, welcome everyone to our next in our weekly series of GREG seminars, the Graduate Research Excellence Grants given by recipients. It's my pleasure today to introduce Shelby Tisinai. Hopefully I got that correct. Shelby's in the final stages of a PhD in plant biology in the Busch lab at Washington State University. I'm told she originally hails from California, but did an undergraduate degree in Illinois. Obviously now is in Washington. And her research today addresses the causes of local adaptation. And today, I believe she's going to be telling us about some work she's done on the role of gene expression changes underlying local adaptation in populations of Buschera stricta along an elevational gradient in the Rocky Mountains. I see that Shelby also this year won an American Genetics Association grant, so congratulations on that, or award. So I look forward to hearing the talk, and I'll just remind everyone that the seminars operate under our standard meeting code of conduct, which will be posted in the chat, and so we expect everyone to follow that. And I will turn it over to Shelby.
Great, thank you. And thank you everyone for coming today. I am going to be talking about differential gene expression as signatures of local adaptation in Buschera stricta. So just as an outline for my talk today, I'm going to give some background information just to sort of set the stage. I'll be talking about environmental gradients, the evolution of gene expression, using gene expression and local adaptation to look at the ecology and local adaptation in plants in general, and then using gene expression to look at the ecology and local adaptation in Buschera stricta. And then I will talk about the big question and the hypotheses that drove my research. I will then briefly talk about the methods that I used thus far and the results that I have and the conclusions that I am able to draw with my results. So a central task of evolutionary biology is to understand how organisms respond to the environment and more specifically the climate. And there are many ways in which scientists can get at this big task. But two of the main ways that are in use are using latitudinal gradients, as shown on the figure on the left, from a study from Dacko, Kohling, and Matthews in 2020. They were looking at adaptation to climate in Arabidopsis along its latitudinal gradient. And the other way that is commonly done is using an elevational gradient, as shown in the figure on the right, from Montesinos Navarro et al. In 2010. And they were looking at climatic adaptation to climate and also in Arabidopsis, but along that elevational gradient. Temperature typically varies with latitude, decreasing as you move poleward, and temperature also varies with elevation, decreasing as you increase in elevation. And because of this, oftentimes these two methods, the latitudinal gradients and the elevational gradients, are thought to be two sides of the same coin, and it's typically thought that results that you find on a latitudinal gradient can be applied to elevational gradients. Haltbritter et al. In 2013 found that these do not agree. Latitudinal gradients do not equal elevational gradients. And the reason for that is thought because range limits in mountains are far more gradual. And this is likely due to the shorter geographic area that you span in mountains. So isolation by distance is much lower in mountain settings than it is on latitudinal gradients. However, changes and fluctuations in climate are more closely tracked in mountain settings. So I want to talk a little bit more specifically about mountain ecosystems. That is the method that I used in this study. There are a few environmental factors that co-vary nicely with elevation. The two big ones are CO2 partial pressure and temperature. And I'll speak a little bit more about temperature. For every 100 meters of elevation gain, you lose 0.4 degrees Celsius. And since 100 meters of elevation gain really isn't all that much in terms of geographic distance, this can equate to being a very strong driver of selection and creating distinct life zones or elevational belts within mountains, as depicted on this figure on the screen, with grasslands and foothills occupying the lower parts of the mountains. It's much warmer down there, and going all the way up to the Alpine and the Naval, where it's much, much cooler. Other environmental factors that we typically think of in mountain ecosystems, such as precipitation and even to some extent UV radiation, are a little bit more of a mixed bag. They do co-vary with elevation, but it's not quite as a nice covariance as temperature and CO2 partial pressure. For example, light radiation, it does, as I said, it does co-vary with elevation, but it does also depend on the aspect of the slope that you are on. If you're on a northern facing slope, then you're going to be getting less UV radiation, less light radiation than you would be if you were on a southern facing slope at that same latitude or at that same elevation. Soil properties also are one of these sort of mixed bag environmental factors. There are some altitudinal mediation effects going on with soil properties, but bedrock is the predominant driver of any changes in soil properties. So why should we use gene expression to understand how populations or how organisms respond to their environment? If you do not use gene expression to look at population differences, you may underestimate differentiation and miss potentially adaptive responses, which we would just be underestimating how things are different. And this is particularly important as the frequency and severity of novel environments increases under climate change. It's good to know what's going on out there. Sort of along those same lines, using gene expression to understand how populations vary along or across environments allows us to really get at what it is about the environment that organisms are dealing with. How are they responding? That's why I'm using gene expression to get at these questions. Here, I'd like to just put my study into the larger picture of how phenotype relates to the environment. In the leftmost panel, panel A, environment has a strong impact on the phenotype. So those two lines represent two different genotypes. And in environment 1, both genotypes have a very similar phenotype. They both have green leaves. And as you move to environment 2, the phenotype changes to red leaves. In panel B, the middle panel, the genotypes of the two individuals strongly influence the phenotype, and that phenotype does not change even as the environment changes. In panel C, the rightmost panel, genotype and the environment have a strong influence on the phenotype. This is G by E. I'm not going to talk about G by E. My study most closely aligns with panel B. However, I look at phenotypic differences in a single environment. So there's not really environmental change. And that brings us to constitutive gene expression. Constitutive gene expression is expression that does not vary despite environmental change. It's genes that are just sort of always turned on at the same level. And in a population, constitutive gene expression could arise due to drift. Random changes, random reproduction can lead to constitutive gene expression, and this, at a biological level, this would look like no or very low correlation to common drivers of selection, such as temperature. Gene expression may also remixation or become constitutive through selection, and gene expression plasticity is very costly. So constantly diverting energy to changing your gene expression profiles um, can help you survive, but it will also limit your ability to reproduce. So under scenarios of expectable environmental conditions, it may be advantageous to have some genes always at the ready, so to speak, sort of in anticipation of these predictable environments. And there is theory and empirical evidence that suggests that genes relating to fitness, life history, and general plant function would be the genes that would be most likely to have constitutive expression differences among populations. So that means that using divergent constitutive expression or looking at divergent constitutive expression among populations can provide a metric with which to assess recent selection on response to the environment. And a common way to look at genetic divergence is through the metric, the FST metric, that compares the amount of genetic variation among and within populations to the total amount of genetic variation within the entire system. And departures from normality in this FST distribution are often interpreted to suggest selection. However, most of the traits that would be advantageous to have stabilized expression are polygenic or quantitative. And FST is a poor metric to use to assess selection on polygenic traits, since it doesn't really account for the additive effects of many genes on a trait. So in 1993, using Wright's 1951 work and Landy's 1992 work in theoretical population genetics, Spitzer used a metric he coined QST to measure divergence in polygenic traits. And under genetic drift or the null model, it is theorized that the distribution of QST will be approximately equal to the distribution of FST, as shown in the figure on the screen, taken from a paper by Michael Whitlock in 2008. Like FST, deviations from this distribution can implicate selection, directional selection when QST is greater than FST, and balancing selection when QST is less than FST. There are many, many studies that look at phenotypic and gene expression differences along environmental gradients in many different plant genera. For example, Engert and Shimsky in 2005 used Mimulus cardinalis and Mimulus louisii to look at phenotypic changes along an elevational gradient. Couriers et al. In 2014 used Mimulus guttatus. Adrian and Shimsky in 2012, along with many, many, many others, have looked at phenotypic and gene expression differences in Arabidopsis thaliana, both along latitudinal and elevational gradients. Hall et al. In 2007 used QST-FST comparisons to look at or to implicate selection or phenotype using Populus tremula or European aspen. And very recently, Yi et al. In 2023 looked at differential expression along an elevational gradient in a rhododendron species. So here I would just like to introduce my study species just to get a little bit of a closer look at Bucharest stricta. Bucharest stricta is an emerging model organism used to study ecological genomics. It occupies diverse habitats. It can be found in arid and rocky scree environments and going all the way to moist alpine meadows. So it occupies a very strong elevational gradient. You can find it in the foothills and also all the way up to the alpine. It also has utility for latitudinal studies because it's common in the Western US, the Northern US, and throughout Canada. It has white to pinkish purple flowers. It's a pretty cute little plant. And it's actually been found that flower color relates, it varies along an elevational gradient as it relates to soil composition and herbivory. It is predominantly inbreeding with FIS values of 0.89 And the populations that I used in my study have FST values that range from 0.26 to 0.53. So it's pretty divergent. It is diploid and it has 7 chromosome pairs. Its genome size is 216 megabases. And what's also really nice about Bucca restricta is that it's closely related to Arabidopsis thaliana, which allows me to do comparative genetics. There has been lots of work done in Bucca restricta looking at phenotypic differences along an elevational gradient. In 2018, Lajimar, McTavish, and Anderson performed provenance trials of Bucca restricta along the same elevational gradient that I've been using. And they found that despite there were weak allelic signatures of local adaptation, but they did find significant differences in fitness metrics, such as seed set and germination success among populations. And the study was looking at phenotypic plasticity, but I think it is really interesting in this figure in the purple box. Within that generation, you do see significant phenotypic differences. That was not the only study done in Bucca restricta. This is just a subset of all of the work that's been done so far. Lee and Mitchell-Olds in 2013 performed QST-FST analyses using Idaho populations of Bucca restricta, and they found that QST was greater than FST, implying directional selection on traits associated with water availability. There's also been a lot of work done by the Anderson group, and they again found that germination success and seedling establishment and flowering time covaried with elevation. And Veda et al. In 2018 found that flower color, herbivory, and drought tolerance covaried with the environment. The studies with the asterisks are studies that use the same populations that I am using in my study. So that brings me to the central aim and hypothesis or hypotheses of my study. So my big aim was to determine the importance of gene expression to local adaptation in Bucca restricta. And my first hypothesis was that constitutive gene expression will differ among populations. Some populations will have some genes that are turned on all the time, and others won't. My second hypothesis is that genes contributing to population differentiation will be predictable based off of the environment and what we know about that system and what has been previously found in the other studies. So I was expecting genes to have functions in thermotolerance, UV response, herbivory, flowering time, all those things. So these are my study populations along the elevational gradient. These populations are from they are established gardens near Rocky Mountain the Rocky Mountain Biological Laboratory in Gothic, Colorado. So it's sort of the southern Rocky Mountain area. Population A is the lowest population at about 2,500 meters above sea level, and population E is the highest population at about 3,300 meters above sea level. So I span an elevational gradient of almost 1,000 feet. I think it's more like 800 or 800 meters. So it's a pretty steep elevational gradient. So I used these 5 populations, and I had 3 maternal families that I was sampling from. The Anderson group were the ones who went out and collected all of these seeds. So they sent me F3 seeds from their lab at University of Georgia. I germinated these seeds using the same protocol that the Anderson lab uses, using wet filter paper, where I put the seeds on wet filter paper in a sealed petri dish and left them in a drawer at room temperature for a few days. And as soon as the radicals emerged and the cotyledons came out and started greening up, I moved them to soil. I just used normal potting mix. And I put them in a common greenhouse setting for 3 months. And these greenhouse settings were the same settings that the Anderson group has been using for the previous generations. After about 3 months, I moved the plants to a growth chamber. They were in the dark for 7 weeks at about 4 degrees Celsius. And this was just to simulate the winter season that these plants are typically accustomed to. After 7 weeks, I moved them back to the common greenhouse using the same greenhouse settings. And I left them there for 3 weeks to sort of recuperate after their time in the dark and to start setting out new leaves. So after about 3 weeks and before the plants started bolting, I harvested new leaves from the plants and I used those to extract RNA. So I flash froze them and kept them at minus 80 degrees C until I was ready to perform my RNA extractions. I used the QIAGEN RNeasy plant mini kit and I followed manufacturer protocols to extract RNA. And I determined the RNA quality using a nanodrop, aiming for nanodrop values of greater than or equal to 2. I then sent those samples off to for total RNA sequencing, and I expected a sequencing depth of 30 million line reads and greater than 30 nucleotides in length. So once I got all those data back, I aligned them using star aligner to the Arabidopsis thaliana reference genome. And then I put all those data into feature counts. And feature counts generates a matrix of raw expression values that can then be pulled into DE-seq2. DE-seq2 normalizes the data to read counts per million and then performs differential expression analysis among sample groups, or in my case, among pairwise populations. Using the normalized data, I performed a QST-FST analysis to determine if there were signatures of selection on gene expression. Using the data set of differentially expressed genes, I then asked the question, do differentially expressed genes correlate with elevation? And to do this, I used a weighted gene correlation network analysis, or WGCNA. And WGCNA soft clusters genes into modules, and the genes within these modules have similar expression profiles. So WGCNA assumes that these genes within a module have similar function. To further verify that any genes that correlate with elevation have a putative function that makes sense in the environmental context and maybe agree with what previous findings have found, I performed a Go term enrichment using Metascape. And within Metascape, I focused my search on the biological process Go terms. So starting off with my QST-FST analysis, I found across the board that that these ratios indicate that there is directional selection on gene expression. Among all pairwise comparisons, QST exceeded FST. I am still working on the statistics for these analyses, so I can't speak to whether or not these departures from normality are significant. It's also important to note that these ratios represent the average QST-FST ratio and are not loci specific. I do expect that loci that are differentially expressed among populations to have more extreme QST-FST ratios. Those analyses are still just in the works. But nonetheless, these results hint that there is probably something more than just drift happening in this system. So after that, I pulled the data set of differentially expressed, or I looked at the data set of differentially expressed genes, just to see if there were differences among the populations. And I found that there is variable and pronounced divergent constitutive expression among these populations. This is a volcano plot, the plot on the upper left. On the x-axis, it is the log2 fold change. So positive values indicate upregulated genes, and negative values indicate downregulated genes. On the y-axis is the negative log 10 adjusted p-value so that we can actually see what's going on. It's not super small. Each dot represents a gene, and the red dots are the genes that are not differentially expressed among these populations, or sorry, are not significantly differentially expressed among these populations. And the blue dots are the ones that are significantly differentially expressed. And something that is interesting to note is that the populations that are closer together, both in terms of geographic distance and elevational differences, have fewer differentially expressed genes than the populations that are both further away from each other geographically and elevationally, as represented in populations A and B on the table, with 53 total differentially expressed genes, and populations A and E, which have a total of 407 differentially expressed genes. So after that, I took that big data set of differentially expressed genes and asked the question, do these genes correlate with elevation? And so prior to doing this whole WGCNA analysis, I performed a principal components analysis just to verify that elevation actually was a strong driver of, or a strong environmental factor in this system. So I pulled some important environmental variables, such as elevation, slope, aspect, soil water availability, light radiation, and maximum vapor pressure deficit into a PCA. And I found that elevation had the greatest loading for PC1. So while PC1 is sort of a combination of many different environmental factors, elevation was the most important. So I used PC1 in the WGCNA. In total, WGCNA found 64 modules, and of those 64 modules, 8 had significant correlation with elevation, with p-values less than 0.01. In the figure on the left, which is the output from WGCNA, the y-axis is the randomly generated color designators the WGCNA assigns to the modules. And the bands within the plot, the darker ones indicate greater correlation with PC1. And the numbers within each band represent or are the R-squared value, with asterisks indicating the ones that have p-values less than 0.01. I was curious about what the expression profiles of these 8 modules look like, the average expression of these modules. So I plotted them, and that's the figure on the right. I couldn't use PC1 as the x-axis on this plot, so I used elevation since it had the greatest influence over PC1. So you will notice that many of the expression profiles don't necessarily perfectly align with elevation, but there are some general upward and downward trends represented here. But deviations from that nice linear correlation with elevation are probably due to the other environmental factors that are contained within PC1. So after that, I took all of the genes that were in all of the modules. I just put them all into one big data set, and I performed a gene ontology enrichment with Metascape. And Metascape found 11 enriched categories within these modules. And so that's great. I just wanted to make sure that the expression profiles of each of these genes sort of looked like they correlated with elevation. And I think by and large they do. I do sort of see downward and upward trends depending on whether or not these genes are upregulated or downregulated. The y-axis on each of these plots is the z-score of these genes, and elevation is on the x-axis of all of these plots. So while there is some variability in each of these Go terms, there are some significant or some interesting trends that you can see in these gene expression profiles. As I'm sure everyone here is aware, Go terms don't really tell us very much about what things or how things relate to the environment. So I did a literature search on all of these genes to see like what function they are really playing in plants, and then also looked at it. I looked at how those functions relate to the environment. So circadian rhythm can correspond to the growing season and the flowering time. Growing season changes with elevation. The tops of mountains have a shortened growing season compared to the bottom or the lower elevations. Plants at the tops of mountains flower later in the year than plants in the lower elevations. And as you can see in this table, a lot of the environmental context relate to important things that are found in mountains, such as temperature differences, photooxidative stress due to high light and CO2 partial pressure, pathogen response. There is some evidence that pathogen intensity co-varies with elevation, and so on and so forth. These highlighted terms, I just pulled them out because they prevent clear implication of the environmental drivers that might be driving divergence in this system, whether it be biotic or abiotic. And this is just due to the seeming pleiotropic nature of these genes in these categories. But it is important to note that these enrichment categories do relate to environmental conditions that are important in mountain ecosystems and can co-vary with elevation. So what does it all mean? In sum, I did find evidence of selection on quantitative characters in my QST FST analyses. Again, I'm still working on the statistics of it, but QST does exceed FST in all instances. And so this suggests that there's something more than drift happening. Further implicating selection or something more than drift is that there is constitutive gene expression among plants from different populations as grown in a common garden setting. And also interesting is that these differentially expressed genes co-vary, they have a strong correlation with elevation. And the putative function of these differentially expressed genes agree with previous findings and make sense within the environmental context. Some future directions that I think would be really interesting to take this work in, would be to do experimental evolution in a greenhouse to try to untangle these dual responses of those highlighted terms or of the genes contained within those highlighted terms from the previous slide, just to further clarify the driver of selection. And this can have different implications under climate change, whether it be biotic or abiotic. I also think it would be really fascinating to look at gene expression in multiple environments. So gene expression plasticity and using that as a quantitative character to better understand how gene expression plasticity varies along this environmental gradient. So with that, I want to say thank you so much for listening to me talk about my research today. I really appreciate the opportunity. I also want to thank the members of the Busch Lab, my advisor, Dr. Jeremiah Busch, and my lab mate, Henry Landis. They're both wonderful people who have listened to me talk about this probably far too much. And also the past members of the lab, Dr. Evan Hiltman, Carly Pryor, and Thomas Buchlow, they have also listened to me talk about this probably till their ears fell off. A big shout out to the greenhouse manager, Amanda Linsky here at Wazoo. She lets me grow odd non-model plants in her greenhouse that require all sorts of environmental conditions. A big thank you to my collaborators, Dr. Jill Anderson and Dr. Derek Denny, and a big thank you to the Society for the Study of Evolution for the money. I could not have finished this project or gotten as far as I did without those funds. So thank you. With that, there's some references.
Thank you. Fascinating talk. We have some time for questions. People can feel free to just put up their hand and I can call on you and you can unmute and ask it, or you can put something in the chat and I can do my best to read it out. I see one there. Minoko, go ahead.
Yes. Thank you for your interesting presentation. And yeah, you focused on the influence of you partly focused on the influence of elevation on the gene expression this time. And I'm curious about whether the global warming had an influence on gene expression, of the plants in in the high elevation.
Yeah, so how might climate change be impacting those higher elevation plants?
And do you have any kind of like your idea or some kind of data which like suggesting the global warming has an influence on the gene expression or don't have so much influence on the gene expression?
Yeah, I haven't collected any data looking at gene expression or how climate change has impacted gene expression in mountain ecosystems. But there have been a lot of studies looking at phenotypic change under climate change. Like, for example, in Buca restricta, in the system with these same populations, flowering time has advanced. So that implies that there probably is some changes or there have been some changes to gene expression for flowering time. That would be a really fascinating thing to look at to compare how that might be changing. Did that answer your question? Kind of.
Yeah, thank you.
Maybe. So I'm curious, in the further direction, you note that it'd be interesting to do some greenhouse studies to try to get at the drivers of selection. And, at first thought, that seems great. Like, it'd be nice to know what the ecological factors are that are driving local adaptation. It seems to me that elevation may be a particularly challenging one because do you think it's actually likely that there's a single dominant factor here that differs among these, as opposed to, like, as you know, changes in elevation correlate with changes in all sorts of biotic and abiotic factors. And I start to worry that if you pull one of those out and manipulate it in a greenhouse, even if you do find something, its contribution may be one small bit of a whole lot of drivers, and its contribution may actually differ depending on the values of those other things, which you're presumably holding constant. Like, there's all sorts of trade-offs in how they respond to. There's probably dozens of things that vary with elevation, and how they respond to one in isolation may be very different to how they respond to one when a whole bunch of other things are varying, which makes it a very daunting problem. And maybe we shouldn't, worry about that. And you got to start somewhere. But is this
right?
Something you think is actually doable?
That's a great question. Really, mountain ecosystems are really just like, in all ecosystems are a mixed bag, right? Everything has 1.5 million different things happening that influence how a plant might respond. But you're totally right. Elevation that's like, what is elevation in terms of biology? It's sort of the culmination of many, many different effects. I think what that greenhouse study might just get at is for each individual population, what might be the prominent driver that is leading to population differentiation. However, as you said, there's so many different things that can push populations to diverge. It's also important to note that this is very local specific, right? What's happening in this region of this part of the Rocky Mountains could be very different in what's happening in another part of the Rocky Mountains, and very different than what's happening in a similar environment in the Himalayas and in the Alps and the Andes, so on and so forth.
It's interesting, like when you do this TCA, and elevation comes out as being a dominant contributor to PC1, one way to interpret that is sort of like, we haven't measured the environmental variables that elevation is most strongly, like we're missing some key factors here that are different among elevations. You almost want to do a discriminant function and have elevation as the grouping, and then say, all right, what biotic environmental variables are differing the most, and then start, I guess, with the assumption that things that differ more are more likely to generate strong selection. But anyway, this sounds more negative than I intend it to be. It just seems like it's a fascinating sort of question: is there actually a single driver, or is it a quantitative thing with dozens of differences that are all contributing, in which case isolating particular ones is going to be really difficult. Anyways, I'll leave it there. I see Jeremiah has a hand up.
Yeah, I'm going to show my ignorance about my own student's project here with my question. But Shelby's used to this. So you have this list of interesting genes that, their expression varies. And I was wondering if the Anderson lab had done another analysis looking at which genes are the most divergent in the genome and whether they overlap at all. Has that been done? That's just me again showing my ignorance.
As far as I know, that has not been done. That is actually something that I am hoping to do, because I want to look at QST and FST on a loci specific, what's the word I'm looking for? I'm taking a high level of QST, FST, and I want to get more specific. So hopefully with that analysis, I can actually identify those loci that are more divergent among these populations.
Do you have, I don't think I missed anything in the chat. Nope. Are there any other questions that among the audience?
Yeah, I just wanted to ask, the plants were in the dark for 7 weeks at 4 degrees to simulate winter, but
Yes.
but like the sun is still there sometimes in the winter,
They're
right?
under snowpack.
Okay,
Sort of
thank you
assuming
for
that they're under
explaining.
snowpack. Yeah.
Okay,
Yeah.
didn't know.
No, that's a good question. And they could have some light, but the protocol that the Anderson lab has done in the past, it's been like an incandescent light bulb if there is any light when it's fertilizing. So I just figured I would save on some electricity and just keep them in the dark.
I see, and that's true for even the lowest elevation, that they'll be just buried.
Yeah, especially in this region of Colorado, near, so I don't know if you're familiar with Crested Butte or Gothic, but they get tons and tons of snow, like sometimes upward of like 25 feet of snow in the wintertime. So these plants are buried.
Yep, got it.
I've got a colleague that's worked up there, Jessica Forrest, so I hear all about it. I see a question in the chat. Since elevation is correlated with climatic variables, have you tested these factors? So presumably climatic variables in relation to your differentially expressed groups.
Sorry, let me see that. So as in like, have I tested each individual climatic variable with the differentially expressed genes? I believe I have actually. I think summarizing them under PC1 and under elevation sort of does summarize what each of these are looking at. I don't know if that answers the question.
Yeah, I'm not sure. We can
Uh,
always,
yeah, we'll see if Masi responds.
are there
Um,
any other questions while we wait? I
yeah,
see Thomas has got a hand up there.
Yes.
Yeah, go ahead.
So I was thank you for your talk, Shelby. I've been waiting to see the culmination of all these projects. But I was thinking about I maybe missed the first like 5 minutes, and maybe you said this and I just didn't pick up or I just missed it. But constitutively expressed genes just means genes that are turned on all the time, but they could still like my understanding was that they are still like plastic. They can like change if you put them into different environments without change in genotype. And so did again, maybe I just missed this, but is it possible that a lot of this variation that you're seeing in the constitutively expressed genes is just plasticity across the elevational gradients? Or did growing them in the greenhouse show that no, that those genes are expressed at the same level in the field and in the common garden?
Mm, that's a good question. These seeds were F3 seeds, so they had been grown in the greenhouse for
Oh,
for three
okay.
generations. So um, if there were plastic differences coming from like maternal effects, hopefully those effects would have been reduced.
Right.
There definitely could be some variation due to greenhouse settings, right? I'm growing them in a different greenhouse under the same conditions, but a different greenhouse than what the Anderson lab has been growing them in. So there could be some differences there. But because I kept subsetting this gene set down to be correlated with elevation, and because these genes were being turned on in an environment that was not their elevational home, I feel pretty confident
Yeah.
saying that there's something going on.
Yeah, for sure.
I guess the ultimate study would be to do this like 12 times over, take all the genotypes and grow them at a set of 5 elevations, maybe at multiple different sites. So it's an elevation effect, not a site effect. And it's like 12 times the size of this or something. Some trivial
Yeah,
little study like that.
some trivial little study. Yeah.
Yeah.
Yeah, I know the Anderson group has done something very similar to that. They did do a little bit of genetics, or like, yeah, they were looking, I think, at allele frequencies. And they did provenance trials. So they did, move the plants around from their home into different environments. But it would be really fascinating to look at it from a gene expression point of view.
I guess the difficult challenge on getting, if you start transplanting plants into the wild and growing at different elevations, then, you sort of want the average effect of growing at that elevation across many years. But any given year, weird things happen. Like climate isn't the same at 1,000 meters every year. And so it gets really
Right.
challenging to try to, under natural, semi-natural conditions, you don't get the average effect of the climatic variables that matter. You get the effect of that particular year, which, makes it really
Right.
hard, probably. Yeah.
Yeah.
Any other questions from the audience?
Yeah, I just wanted to ask, I don't know if anybody else had this question, but so what were the genes that made up those modules with the highly significant p-values? I was
Yeah. So I
yeah.
will move over to this
Maybe a
slide.
long
Um,
list.
it is a long list, and I don't know if you can really see that. They're pretty tiny. I was trying to fit all 11 um, all 11 plots on the screen. But it is a long list. I think how many genes were there? I think there were greater than 75. So not as many as other studies have found, because I kept subsetting them down to make it chewable. But nonetheless, it is a big list. So
Yeah, so how do I
and
tell
yeah.
what gene it is?
Sorry. These gene or each line corresponds to a gene. So the gene ID is listed in in the key on the side of the graph.
Right, but these are like your values, right? They don't have like meaning outside of your analysis. Is
Oh,
that
no. So you can take these gene IDs and plunk them into TARE, and TARE will actually tell you what they are. These are the TARE IDs.
right. And so these are all from which module?
Great question. So these are all of the modules considered. I took the gene set out, so they were irrespective of module.
Right.
It
So
was
my
just, yeah.
original question was like so if they're from any
Oh,
kind of module, then they could be from a module that doesn't correlate with elevation. They could be from a module that weakly correlates with elevation. They could be from one of those purple modules or highly orange modules that strongly correlates with
yeah.
elevation. So I'm wondering what were the genes that were assigned to the modules highly correlated with elevation?
Gotcha. So it's all of these. I only took the genes in those highly correlated modules and put them into the GO term enrichment
These are the
analysis.
11 modules that correlated, that showed the strongest elevational, yeah, of all.
I
You had
see. So
63 modules or something total,
I
right?
see. So these are the 11 most significant modules. So this is like all the genes from those 11 most significant modules, right? So then
Yeah,
like
yeah.
so then what are some of these genes, since I don't recognize AT AT1G, it doesn't.
Yeah. That's another great question. Um,
That comes from
they are,
Arabidopsis, does it? That data? Yeah.
yeah, yeah. So these, since I aligned to the Arabidopsis genome, I was mapping and doing comparative genomics with it. So these are Arabidopsis genes. Um, I don't know the names of the human names of these genes. Um, but if you were to take any one of these genes and put them into TARE, you can see all of the list of publications that have published on what this gene is associated with, the function that these genes are associated with.
Did any genes appear in multiple modules among these 11?
Yes,
The same genes
yes,
appear? Yeah.
yeah. So I had 8 modules from WGCNA, but 11 functional Go terms. So there is some overlap. There's
So
some pleiotropy going on. Yeah.
right, Howard, I think a gene can be assigned to multiple Go terms, but a gene cannot be assigned to multiple modules. I believe that's
Yes.
correct.
Yeah. Okay. Yeah. That makes sense. Yeah. Cool.
Yep.
Well, we'll look forward
Okay, thanks for explaining. Yeah.
to reading this when it's published.
Yeah, thank you. I'm excited to having it published.
Well, maybe join me in one final thank you to Shelby for an interesting talk, and hopefully we'll see some of you next week at next week's talk.
Yep, we'll see you guys next week for the final Greg seminar. Wow. It's been quite a journey. Yeah, thanks so much, Shelby. See you
Thank
guys
you.
later.
Take care.